Hello and welcome everyone to today's presentation on IMDA systems for instantaneous data-rich applications presented by Scott Morris. Scott Morris is currently the Applications Engineering Manager at Azbil BioVigilant, Inc., a company focused on delivering real-time detection technologies to empower users in the pharmaceutical and life science industries. Scott graduated with a degree in optical science and engineering from the University of Arizona and has been with Azbil BioVigilant for 6 years. Before we get started, we'd like to announce that we will be taking some Q&A at the end of today's presentation. If you have any questions, please send them via the Q&A function to the panelists using your webinar control panel, and we'll answer as many of them as time allows for. And with that quick introduction, thank you again everyone for joining us. And Scott, I'll hand the floor over to you to begin today's presentation.
Controlled environment within the pharmaceutical and life science industries. And then, of course, we'll have the Q&A session at the end, and you can ask everything that I forgot to mention. So first things first, what is an RMM? Simply, it stands for Rapid Microbiological Method, and it's any instrument or method that generates microbiological data faster than the traditional growth-based methods. Typically, those are going to require 3 to 5 days for your standard growth-based sample, but of course certain tests like a sterility test could take up to 2 weeks. So RMMs can offer multiple benefits to their users. Some of those include improved speed, performance, such as accuracy, precision, improved cost savings, and quality. And RMMs can be key tools to help enable a paradigm shift from batch to parametric release. And for those of you that may be unfamiliar, batch to parametric release basically is a paradigm whereby instead of having to wait for sterility samples to come back from your QA department before you can release a batch of product, if a firm is able to demonstrate through sufficient process controls that the product was produced with acceptable quality through all stages of the process, they could use parametric release and release that product as soon as it comes off the manufacturing line. So for many large and global pharma companies, this is something that they have been investigating and pursuing for a bit of time now. This paradigm shift is also supported by several regulatory and quality initiatives around the world. Here domestically, the FDA encourages rapid micromethods through a couple initiatives. One of them is PAT, which stands for Process Analytical Technologies, and also the QBD, which is Quality by Design initiatives for continuous online monitoring of one's manufacturing process. Over in Europe, the EMA supports parametric and real-time release with suitable critical parameter data, one of those facets being environmental monitoring data. Also, we're seeing that a lot of large pharma companies are using internal tools for risk management and enhancing their process knowledge, finding that a lot of value is derived by spending the time and effort to really understand and optimize their process and move from a reactionary type of stance to one that's more proactive. Also, rapid micromethods have the ability to transcend certain limitations of your traditional growth-based micromethods. And a few of those qualities are that in certain cases your results can be generated in real time, instruments can be run continuously, and they do not require sample collection by an operator in the clean room, meaning it could be run remotely. Now, of course, this has the obvious logistical benefit of reducing the burden to collect and process environmental monitoring data, but it also has the added benefit of helping reduce clean room operator presence in the most critical areas where those samples need to be collected. The reason we want to try and reduce the operator presence is because, as is pointed out in United States Pharmacopoeia 1116, it's a recent edition, operators are the primary source of contamination in a clean room. People shed about 10 million skin particles per day, so even the best clean room garments aren't necessarily going to be able to retain all of those. So reducing the dependence on manual sample collection reduces contamination in the first place. Now, I already mentioned that enhanced performance of RMMs may be possible over traditional methods, and they may even have sensitivity down to a single microbe. Now, note that I didn't say a single CFU. CFU is the unit of measure that everybody is familiar with for detecting microbes, but the mechanism for measurement in some RMM technologies is very different than observing an actively growing colony on a growth plate. And this difference engenders one shift from your traditional techniques, which is the ability to detect VB and C, also known as viable but not culturable microbes. So let's talk real briefly about that. I don't have time to cover VB and C exhaustively here, but we can reference some material that's been previously presented at pharmaceutical industry meetings, like this graphic from Dr. Ed Tidswell of Baxter. And basically what he's showing here is a map of microbes across all possible metabolic states and their potential from growth. You have everything from a dead microbe in the lower left corner of his graphic to an actively growing colony in the upper right-hand corner of the graphic. Now, using traditional growth-based techniques, detectability of these microbes is intrinsically linked to culturability. So this green region shows the extent of potential microbes in his estimation that you would be able to detect with growth. So with the exceptions of dead cells and sublethally injured cells over on the far left side of the graphic, the remaining portion is what could be considered viable but not culturable. The last 30 years or so of research on the topic indicates that these VB and C microbes are indeed capable of replication, and they do have clinical significance. This slide from Ian Simmons of GSK illustrates the pervasiveness of the VB and C phenomenon for common human flora, kind of demonstrating that clinical significance. So with standard tryptic soy agar growth conditions, most of the microbes on his chart, as indicated by red, will go completely undetected. So clearly there is a potential and a need to be able to detect microbes beyond those which grow under your standard TSA growth conditions. I'm pretty sure the standard growth conditions he was referencing were 20 to 25 C for 2 days, followed by 30 to 35 for a couple more days. So in addition to improper growth conditions, other common VB and C vectors include damage and environmental stressors like starvation, desiccation, and chemical exposure. Now, if those methods of stressing microbes sound familiar, they probably should, because those are the conditions that are typically found in most clean spaces, particularly those related to the pharmaceutical industry. So many microbes that may exist in these environments are already predisposed to being stressed and potentially not detectable using traditional methods. So let's talk a little bit about RMMs in general. So considering the requirements for culturability is also one way to interpret rapid micromethods. And this slide shows one possible scheme to categorize the many RMM detection technologies. I don't have any identification technologies listed here, but detection technologies available on the market today. So the left-hand side of the graphic, as you can see, are rapid micromethods which are growth or viability dependent. They still require growth, albeit they are able to deliver those growth-based results much faster than the traditional method. The right-hand side of the graphic includes all of those RMMs out there which are able to detect microbes in a method independent of growth or viability. And while this taxonomy certainly isn't exhaustive, you can see that the technology we're discussing today, the IMD system, is a technique which detects intrinsic microbial fluorescence via optical spectroscopy. And of course, because this fluorescence is intrinsic to the microbe, well, we're not looking at growth, we're not adding any chemicals or reagents, this technology falls into the right-hand side of the taxonomy. So let's talk about the core detection principle of the IMD, give you a sense of what this instrument and technology actually is. Within the system, detection of microbes occurs via the simultaneous detection of intrinsic fluorescence and Mies scatter. Now what you're looking at on the left-hand side of the slide here is the core detection engine of the IMD system. So first, how does this system work? It pulls environmental air from the outside of the instrument in through the interrogation zone, where a 405 nanometer laser interrogates 100% of the particles which go through the airflow path. For each particle, two different things can occur. Mies scatter is going to occur for all particles in our size range, whereby the scattered light is proportional to the particle size. And the magenta region, which is being shaded here, illustrates the scattered light which is collected by one set of optics and focused onto the scatter detector. Me-scatter is the technique used by most commercial particle counters in the industry, and probably many of you are familiar with the particle counters you're going to see all over your manufacturing areas and clean spaces. Those are very likely using me-scatter for particle detection. Simultaneously, if the particle contains the metabolic fluorophores NADH and riboflavin, as well as the picolinic acid in the case of endospores, intrinsic fluorescence will be generated and is captured by a second set of optics and directed onto the fluorescence detector. And you can see that fluorescence illustrated by the green shaded region on the screen. These scatter and fluorescence signals, if present, are captured and analyzed simultaneously and in real time to determine the particle size and the biologic status. Since these metabolic fluorophores, the NADH and riboflavin, are universal, this is what enables detection of viable but not culturable microbes. The final overlay we're looking at now is an optical scatter model for the Me and fluorescence channels, as Azbil BioVigilant uses tools like this to ensure we meet customer specifications for the system, like single-cell detection, and we'll talk more about that in a little while. So let me give you a little bit of an overview from the system level for IMDA systems. So we just covered the core detection engine, but at the system level, we offer two different products. Each is specifically tailored to customer requests. Those two models are the IMDA 300 and the IMDA 350, and this table shows some key parameters of interest. Both models use the same core detection platform that we just covered. By design and has been demonstrated through validation, this technique achieves single-cell microbe detection. The major distinction between the two models is the sample flow rate. The IMDA system, IMDA 300 system on the left-hand side of the table, has a flow rate of 1.15 liters per minute and interrogates 100% of that airstream. To achieve the market's request for a 1 cubic foot per minute instrument, and 1 cubic foot is equivalent to 28.3 liters per minute, it's a very standard flow rate you'll see of various instrumentation across the pharmaceutical and life science industries. And so that was a common request among our customers. To achieve that flow rate, the IMDA 350 system incorporates a flow concentrator which uses virtual impaction. This is the technique akin to the physical impaction method for traditional air samplers. So many of you are probably familiar with the traditional air samplers that use an auger plate. It sucks in air, all the microbes and particles get impacted onto the auger plate, and that's what's put into an incubator to try and detect the microbes. So the technique we use is similar to that in terms of impaction. The sample flow rate is 28.3 liters per minute, from which the particles are concentrated down to 1.15 liters per minute for interrogation. While the 1.15 liter per minute airstream still has 100% interrogation, not necessarily 100% of the particles from the higher 28 liter per minute flow rate make it into the interrogation flow. This is the inherent trade-off for achieving a higher flow rate while still maintaining the necessary fluorescence sensitivity. And it's worth noting that traditional air samplers also have this trade-off where they're not able to collect 100% of the particles. And this is not a focus of the presentation, but if there's any questions, we could talk about that at the end. So you see on this graphic here that there's a metric for the IMDA 350 system that's listed as the D50 value. And it says it's less than 2 microns here. The D50 value is a common metric to characterize the physical efficiency, and it denotes the particle size at which at least 50% of the particles are concentrated and detected. Our measurements put the D50 value just under 2 microns, which is performance on par or better than many of the common air samplers in industry today which utilize impaction. A common standard associated with just general particle counting is ISO 21501-4. And this has been out for a couple of years now. Some of you on the call might be familiar with it. So while the IMDA system is first and foremost a biologic particle detector, some customers have expressed interest in how the IMDA system compares to this ISO metric for general particle counting. For the ISO metrics associated with particle detection and counting, the IMDA 300 system meets all of these. Due to the inherent properties of the aerosol concentrator, the IMDA 350 is enabled to meet the counting efficiency metric because, as we just discussed, detection or concentration is a function of particle size. So beyond the particle detection metrics, we're currently working on implementing additional procedural steps that would be necessary to offer a fully compliant calibration of the IMDA 300 system. So, when considering the potential applications for an IMDA system, the user has a choice of models depending on the trade-off between sample flow rate and counting efficiency. We've aimed to design a turnkey system, and then to that end, you can look at the sample environmental specs on the table and see that we've designed a system which should operate across multiple environments encountered within the pharma and life science industries. For example, the IMDA systems can handle various pressure loads from aseptic isolators, which are at positive pressure, to negative pressures found in other clean room spaces, and automatically adjust for the optimal flow rate of each system. Likewise, to ensure the system is operating nominally for these demanding applications, the IMDA has a host of onboard system health monitors, including flow rate, laser, and an automated sensor self-test. Also at the bottom, you can see some of the standards and guidance documents to which we've tested and demonstrated compliance for our system. So on this slide, we can see what is called the PharmaMaster graphical user interface, and that's the software with which a user interfaces to the IMDA system. And this is really the interface that provides the user the real-time and data-rich feedback that this presentation is named after. So what we're watching now is a historical sample playback. And I know it may be difficult at the moment to see that the video is playing, but you'll see action soon enough. And this is an option that can be used for every sample collected, but is particularly powerful for data review and analysis when the optional video module is enabled. And you can see down in the right-hand corner of the screen that video was collected during a sample. And what we're watching is a simple media plating operation being performed in a biosafety cabinet. The isokinetic probe that was used to collect the air sample is visible in the foreground. And you can see on the left-hand side of the screen that there are no particles, biologic or otherwise, detected during the beginning of its test. And in the center of the screen, you can see with green boxes around the zeros the total number of particles that have been detected so far. Now you'll notice in the video that the operator's arm was just exposed when the glove came off. And we can watch and see how the IMDA system responds. So one particle has been detected so far, a biologic, as indicated by red. And then all of a sudden, the IMDA system detected a spike in all particle categories. That's particles greater than half micron, greater than 5 microns, and biologics. But soon after the operator was successfully able to put the glove back over their sleeve, you can see that the environment returned to a zero baseline. So under normal measurement methodologies, perhaps using a settle plate in the biosafety cabinet or any other method, the odds of detecting this excursion within your biosafety cabinet is really low. So you probably wouldn't even have an indication that there was risk to the operation or product that was inside the safety cabinet. So over on the right-hand side of the screen, the PharmaMaster interface, you can see what we call the control panel. And this has various functions that are also very helpful to the user. At the top, you can note that we have continuous and point sampling modes depending on the type of sample the operator wants to take. We also have configurable measurement profiles. And this allows the user to create customized alert and action levels for different applications, as well as defining what we call events. And events are a way to tag samples in the data files, reports, and the audit trail when counts exceed a predefined alert or action limit over a predefined interval. And this makes reviewing excursions from long samples as easy as jumping between tagged events. So if, for example, you were monitoring an 8-hour shift of a manufacturing process, you could set predefined intervals, and then during the 8-hour shift, if any events occurred which exceeded those, while they may not necessarily be your action limit, you'd be able to tag them as events and jump between them during data review. And these are the tools that allow the user to respond quickly and decisively to excursions, as well as meaningfully review the data to distill the process understanding. And that's data that's indicative of microbial control for the entire batch. That's the kind of data that one would want if they were going to move to something like parametric release. From the data collected, the user also has multiple different options for automatic reports and data export. So while the graphical user interface displays the counts from that excursion in a very straightforward manner, there's a whole lot going on under the hood of the software, so to speak. So the data acquisition software which powers PharmaMaster is collecting data at hundreds of thousands of hertz and applying data processing algorithms in real time. Our data acquisition software also includes powerful engineering tools that allow our technical team to fully monitor and test the detection performance of the system. For example, what you're seeing now are a couple screenshots from the engineering module of the software illustrating blower sensor controls and raw waveforms captured for weakly fluorescent particles. So let's talk a little bit about IMDA validation. So rapid micromethod validation, RMM validation, is obviously an important topic for anyone considering an RMM application which falls under regulatory jurisdiction. And in a few slides, we'll talk about some of the different applications and which ones may or may not fall under regulatory jurisdiction. But to support the end users in what's often considered a complicated and expensive endeavor, Azbil BioVigilant completed a formal validation of both IMDA systems, which was a 2 to 3 year process that culminated in 2011 with the filing of 2 Type 5 drug master files documents with the FDA. So the ultimate goal in this validation effort was to provide meaningful and relevant data to the stakeholders demonstrating, quote, that the performance characteristics of the RMM meet the requirements for the intended application in comparison to traditional methods. And that's the goal straight from a standard called USP 1223, United States Pharmacopoeia 1223. And that's the current benchmark guidance document for validation of rapid micromethods in the United States. So this graphic here just kind of illustrates the 6 components that we considered as part of the validation strategy to deliver relevant and meaningful information to the end user, not just data, to both customers and regulatory bodies. While the scope of the validation effort is far too big to discuss entirely in an hour-long webinar, we'll hit on a few highlights, and summary documents of this validation effort with more detail are also available from our website. So let's talk a little bit about the test design and the strategy. So there's 2 different guidance documents out there that are looked to for validating rapid micromethods. Currently in the United States, it's USP 1223, and in Europe, it's EP 5.1.6. There is also a PDA technical report number 33, which is about a new edition is about to be released in the next month or so, which has guidance on validating rapid micromethods. But what we tried to do was integrate the requirements from, USP 1223 and EP 5.1.6 and harmonize them to provide a more holistic and universal approach. And from that integration, we have the 9 basic metrics for comparison, and those are in the lower left-hand corner. So those are accuracy, precision, limit of detection, limit of quantification, linearity, range, specificity, ruggedness, robustness. And those are standard among both guidance documents. So as part of our effort to deliver data that was relevant and meaningful to our customers, for which action levels can be a single CFU in a grade A environment, a fundamental tenet of our testing was to create the most challenging scenario from a detection standpoint. And the way that we did this was using monodispersed microbial aerosols. And monodispersed simply means that the microbes are singly dispersed. They're not agglomerated. They're not in clumps. Clumped microbes are going to have more fluorescence and therefore are going to be easier to detect, but doesn't necessarily support the goal of being able to detect a single microbe in a grade A environment. So we only use singly dispersed microbes. Now, while they may be commonly detected as agglomerates, which means a clump of microbes, or fomites, which means a microbe attached to another particle like a flake of skin, in a natural environment, this may be the case in which they're detected, but published literature confirms that microbes do exist as submicron aerosol particles, meaning that they're singly dispersed. The test metrics for which this type of monodispersed challenge applied were evaluated in a state-of-the-art aerosol test chamber built and validated at our sister laboratory in Japan. So BioVigilant is a subsidiary company of Azbil, and we have a sister laboratory in Japan which helped us perform the validation effort. So within this test chamber, in a little schematic of the test chamber, it can be seen in the upper left-hand corner of the slide that we're looking at here. Within the test chamber for our aerosol validation, we had a bunch of different instruments. We had 4 IMDA systems, 2 of each model. We had 3 reference air samplers. Those reference air samplers were the SAS sampler, the SMA sampler, and the MAS sampler. So anyone who's familiar with environmental monitoring has probably heard of those traditional techniques for growth-based sampling. And we also had a reference particle counter. In this case, it was a Canamax reference particle counter. So to evaluate USP-1223, we tested 6 different microbes at 5 concentrations apiece. At each concentration, we went well beyond the minimum number of required replicates and performed 12 replicates to try and create a large and robust data set. And some of the design experiment parameters are listed in the right corner of the slide here. So speaking of the data set, we developed a monolithic approach whereby most of the USP evaluations were performed on a common data set as opposed to performing different tests for each of those 9 USP metrics. The reason that we tried to approach it with a monolithic strategy is that it's more integrated and precise if you're doing all of your evaluations on a common set of data as opposed to different sets of data. However, to produce this common set of data, it required simultaneous and parallel sampling of all of the instruments I just listed, so all 8 instruments that I just listed, which is not necessarily a straightforward thing to do. But if you're able to accomplish it, it helps preclude bias from inadvertent operator bias, inadvertent bias of the system, etc., and it minimizes systematic error like temporal variation and improves precision. So the scope of this testing was pretty massive. We collected over 1,000 auger plates in the final test, from which over 1,400 comparisons to the 1223 metrics were performed. So let's look a little bit more at the chamber that we used. This is a schematic of the 2.9 cubic meter purpose-built chamber in Japan, which was designed with a specific goal of testing and validating these aerosol rapid micromethods. The chamber itself was designed for compliance with and validated to multiple standards out in the industry, including a couple Japanese standards, 3836, a couple ISO standards, including 14698 and 14644, and several others. So it was validated to achieve ISO 4 particulate levels, which is extremely low. Most clean rooms in the pharmaceutical and life science industries are ISO 5. So it was shown that this chamber could achieve ISO 4 particulate levels and grade A microbial conditions, so less than or equal to 1 microbe or 1 CFU per cubic meter. The chamber contains an aerosol nebulizer in the center, you can see that called out, into which freshly prepared and characterized liquid microbe suspensions were placed immediately before testing. So this nebulizer would create an aerosol, and then all 8 instruments would be able to sample from this aerosol simultaneously. The 4 IMD systems are arranged around the nebulizer, and you can also see, as indicated by the red circles, the 6 mixing fans, which were used to create a homogeneous dispersion of aerosols, and the homogeneity was also validated before the test was performed. The air samplers are operated through a specially designed interface port with automated sanitary valves. In between each replicate, you can see at the top of the chamber, indicated by the green box, there is a recirculating HEPA filter to clean the chamber down between replicates and achieve those very low particulate levels. So speaking of automation, one of the significant and unique things about this chamber is the degree to which it was automated. And as I mentioned earlier, we're trying to use automation to reduce operator error and preclude bias, which improves the repeatability of your test, and it also improves the precision. So once a particular microbe suspension is loaded into the chamber and the test sequence is initiated through software, the entire chamber is controlled by software. Once the test sequence is initiated, the only manual step required during 12 sequential replicates is for the operator to change the auger plate on the air samplers in between replicates. So the system is highly automated, and you can see in this actual photo of the system the interface port for the 3 air samplers, and also some of the computer controls that are used to automate everything. So let's look at some of that validation data that was generated from the aerosol chamber. As I mentioned, the monolithic approach means that most of the USP 1223 metrics like accuracy, precision, limit of detection, and linearity are analyzed from these unified data sets. And what we're looking at here is data from Escherichia coli, a vegetative gram-negative cell, using all 4 IMDA systems and all 3 reference air samplers across 5 increasing concentrations of that microbe. And those 5 concentrations are labeled across the x-axis. T1 is the lowest concentration, up to T5, which is the highest concentration. And the inset micrograph up in the upper left corner shows the microbe suspension before aerosolization. The suspensions were checked visually for particulate contamination and proper cell morphology before they were aerosolized. So each data point on this graph represents the average and normalized counts per liter of air across the 12 replicates that were performed. So 12 replicates were normalized so that it was counts per liter, and then the 12 replicates were averaged, and that's how you get one data point with error bars. You'll also notice that the y-axis of the graph is logarithmic, so that you can clearly see the 5 aerosol concentrations on one graph, and you can see that they span roughly 2 logarithms, a factor of 100. And we found 2 logs was the general limit of dynamic range for the air samplers. So the IMDA system was not the limiting factor in determining the lowest and highest concentration. It was the air samplers. The reason that was a limiting factor is because we optimized testing conditions to avoid air sampler recoveries of 0 at the low end or too numerous to count at the high end. Too numerous to count simply meaning that there's so many colonies growing on the plate you can't distinguish and enumerate them. And that ensures a valid comparison because both 0 and too numerous to count results preclude you from really doing any valuable quantitative analysis. So what we can see from this graph, kind of interpreting it, is that both IMDA models and air samplers show increasing detection with increasing concentration. That's what you would expect, and that's kind of akin to a linearity assessment. So you can see that all instruments are performing somewhat linearly. We also see good repeatability between like models. So the IMDA 300 and 350 have good repeatability among each of the 2 versions of each system. And that's akin to kind of like a ruggedness comparison, showing how closely different models or the same models compare to each other. Both the 300 and 350 perform better than air samplers in terms of normalized recoveries, and this demonstrates accuracy. The accuracy requirement in USP 1223 and EP 516 require that your rapid micro method collect greater than or equal to 70% of the counts from a single reference air sampler. That's worth noting that the requirement for these guidance documents is to compare against 1 traditional method. We compared against 3. And in all 3 cases, you can see that the IMDA systems are collecting higher counts than the reference air samplers. Another general trend that you can notice is the difference in error bars, which is a measure of standard deviation, between the IMDA systems and the traditional air samplers. And this approximates an assessment of precision. So the smaller the error bar, the more precise your measurement is. And you can see that the air samplers, even at the higher concentrations, have some limit to their precision, whereas IMDA systems have a very tight precision, and the precision gets even better as the concentrations increase. So let's look at a little bit more data. Here we see the results for Bacillus atrophius. This is a commonly used endospore for aerosol testing because it behaves well in terms of an aerosol and is robust against stresses of the nebulization process. So we can see the same general trends as before with the E. coli. We have the increasing response. That's basically a visual interpretation of linearity. We see good repeatability between like model instruments. That's kind of a visual demonstration of ruggedness. The recovery of the IMDA systems are above the reference air samplers. Again, that's ensuring accuracy. And the error bars indicate good precision for the IMDA systems. And one thing that's kind of interesting and worth noting is the difference in flow rates between all of these pieces of equipment. So the IMDA 300 systems have the lowest flow rate of any equipment on this graph. They're operating at just over 1 liter per minute. The IMDA 350 system has a flow rate of 28.3 liters per minute, which is a common flow rate. And the other system on this graph, which has a flow rate of 28.3 liters per minute, is the SMA reference air sampler. However, the other 2 air samplers, the SAS and the MAS, operate at 100 liters per minute. So that's roughly 3 times the flow rate of our high flow system, and on the order of 100 times the flow rate of our low flow system. And even given that disparity in flow rates, you can see how the results look in terms of precision and accuracy. So that really was something that we were happy to see that flow rate didn't necessarily preclude IMDA systems from detecting more, especially at the lower concentrations, where it could be a hindrance. So what we're looking at now is a graphic illustrating the strategy that Azbil BioVigilant has developed over the past 5 or so years with both the FDA and the EMA in Europe for validation of the IMDA system. The left-hand side of the graphic is what we call primary validation, and this is a validation that is performed by the RMM supplier and includes all the tests that we just talked about. And what that culminates with, at least in the US, is a Type 5 Drug Master File submission to the FDA, and in Europe, the tests and results are reviewed with regulators but also made available to customers. The other side of this validation strategy is what we call the secondary validation, and that's the validation performed by the RMM end user. So that includes your standard qualification protocols, everything from design qualification through installation and operational qualification, and then of course performance qualification. That's the step at which you're showing comparability between the new method that you want to use and the methods that you have traditionally used. So that's side-by-side testing with the IMDA and your conventional method. So the general applicability of this approach for any given RMM obviously depends upon the specific RMM you're talking about, the application, and the regulatory arrangement that your company may have. But for an aerosol environmental monitoring method like ours, it's generally been regarded as either an in-process control and not necessarily requiring a comparability protocol, which is often a requirement in the US, or a type variation, which is often a requirement in the European Union for implementation. However, it's worth noting that both regulatory agencies have expressed a strong interest in being involved during the validation process and data review before implementation. We've found them to be very welcoming and interested in knowing what customers are potentially looking at in terms of RMM validation, and they're very willing to give feedback and review data and help customers understand whether they're generating data that would be sufficient for any given validation. The other thing that's worth noting is that for that secondary validation that the end user has to perform, it's quite common that some of the protocols necessary could even be offered by the RMM supplier, and that is the case with the IMDA system. For example, the PQ that we have available for customers was developed with the help of an outside statistical expert and has even been reviewed with the FDA. So let's talk about applications, all the different things that you could possibly use an RMM in general for, but in particular the IMDA system. So we already discussed many of the benefits, but you can see there's a lot of different ways that those benefits can be applied in different applications. So those applications on the left-hand side of this slide are ones which are typically associated with the formal validation and regulatory submission, while those on the right-hand side may be leveraged without necessarily as much formality. And by reviewing this list, we can start to see certain applications where different models of the IMDA system may be more appropriate. So for example, the high flow rate and the ability to collect more data faster with the IMDA 350 system is typically more popular among our customers for things like operator training and investigation. However, the low flow rate coupled with high efficiency of the IMDA 300 system is sometimes preferred by customers for critical space monitoring, like manufacturing lines where they want minimal disruption to the flow dynamics and high efficiency. So on the left-hand side, these traditional applications, the ones that are typically considered to require formal regulatory submission, it's worth noting that we have leading top 20 global pharma customers around the world that are in various stages of evaluation and validation with the IMDA system. And it's also worth noting that kind of on this left-hand side where you're monitoring controlled and classified environments, that there is a leading U.S. compounding pharmacy which has been evaluating the IMDA system and is now in the process of implementing in a new site. So on the right-hand side of the slide, we have some applications that may be less obvious to someone thinking about a system such as the IMDA. So one of those is investigations. Basically what we're talking about here is the ability to use the IMDA to quickly get to the root cause of an investigation. For example, you find that you have higher than normal microbes coming back on some of your settling plates or active air samples in a clean room, but you can't figure out where they're coming from, and you have to perform an investigation. The IMDA system is going to give you that real-time feedback, and especially when coupled with the video, it allows you to help diagnose the root cause rather quickly. And among the several large pharma customers that have used that in the last 12 months to try and diagnose issues that they found in their clean rooms, they are finding root causes with the IMDA system that in the past took them several weeks or months to get to the root cause of. So some customers have found that an extremely useful application. There's also operator training where you can use the instantaneous feedback to demonstrate to your clean room operators the importance of aseptic technique and to use it as a support tool in training qualification. There's also a couple other applications here, one of them manufacturing and maintenance activities. Basically what that is using a system like the IMDA to ensure that you have control of your classified environments even in between performing manufacturing of product. So during plant construction activities or during installation and recalibration of equipment, you don't necessarily want your environment to go out of control because that could present risk when you try and come back under control and produce product. So something like the IMDA could be used to monitor and ensure that you're maintaining control. And then a rather new application that we've been investigating is energy reduction, using something like the IMDA to demonstrate that you're achieving the minimum HVAC levels to control your environment. Many companies simply put their HVAC systems well above the minimums required in various regulations because they have no way to know whether using the prescribed flow rates in their HVAC system ensure control. But if you have a system that gives you instantaneous feedback, you can use that data to demonstrate control. And recently a global pharma company used the IMDA to demonstrate that they could save up to $120,000 a year in HVAC expenses by reducing the airflow. Also, the asterisk indicates that published literature in the form of a case study data and or application note is available for these different applications. So let's get into a case study. This case study was performed by IBSA Pharmaceuticals in Lodi, Italy, and basically IBSA used multiple IMDA systems to monitor critical points in a newly constructed aseptic fill operation during their initial validation. So among several points monitored that, in their facility, we're showing here the two that they considered to be of highest interest. This is the pass-through interface between a grade C and grade A room to ensure good control around the interface and assess the risk of any crossover contamination from C to A. So the left-hand graphic is an image of the grade C side where they used an IMDA 300 system to monitor. And the right-hand side is a graphic showing right on the other side of a pass-through the grade A environment where they used an IMDA 350 system to monitor. So on the left-hand side in that gray C area, what we're looking at is equipment that was automatically removing plastic wrap from tubs containing vials that were to be transferred through the pass-through and then filled in the grade A filling operation. So during testing, they were still trying to optimize manufacturing parameters like the suction of the automatic bag opener. So we could consider this data to represent the worst case scenario, whereby the equipment wasn't finely tuned and manual operator interventions were commonly needed. And IBSA actually desired this because they felt like a worst case scenario enabled a more robust determination of potential risk to their grade A area at that critical interface point. So we can actually watch some video now generated by the IMDA 300 system. So again, we're looking at a historical sample from all the testing that was conducted of the automated bag removal equipment. And you can see in the video that there's bags or tubs coming down the line, and then a tray comes over them and removes the bag and then pushes it through that interface from the grade C to the grade A area. But during this process, sometimes the plastic bag gets stuck and requires a manual intervention to remove. So what happens, you can see it right now, an operator enters from a grade D area, even less controlled area, to perform the manual intervention and clear the line. But what we can see from this data is that while a few particles were detected, and you can see right now in the IMDA system we're getting a very small spike of counts. Overall, considering that the operator was coming from a grade D area, there's very good control and overall very low counts even during that manual intervention. And this data was used as part of their risk assessment for the grade A area. And as I mentioned, a lot of data was also collected in the grade A fill line, right on the other side of that tub pass-through. And they used the IMD 350 system, but it turned out to be really boring data because it was all zeros. So that was what they were able to use to demonstrate that despite an operator coming in and performing a manual intervention, the risk that was being posed to their grade A fill line was quite low. So I already covered most of these conclusions, but I have them here listed for your benefit. Lots of data was collected in grade C and A areas used to support a risk assessment. One of the things that's worth noting is that with highly sensitive equipment like the IMDA system, there can be a potential for non-biologic particles to fluoresce. But the low counts in the grade C area with intervention from grade D confirm that this type of interference is not a problem in their environment, which was good to see. And IPSA considered these tests to be very successful. The company plans to implement the technology as part of their quality by design and process analytical technology initiatives after the fill operation is fully validated. So to kind of wrap up, we have two different products, the IMDA 300 and 350 systems, that are tailored to meet needs for the pharmaceutical and life science applications. In our experience working with customers, different applications and environments are unique. So really to make those applications successful, in situ testing and support is really important. During that testing, the data-rich feedback and the software tools that we looked at coming from PharmaMaster allow the user to distill the relevant and actionable knowledge to incorporate this type of paradigm-changing equipment into their environment. And though we've been collaborating with customers and regulators for several years to bring the current products to market, we continue to advance both our PharmaMaster software platform and the IMDA systems to meet your requests. It's also worth noting that we're working to develop a water-based version of our IMDA technology. So all of these projects are consistent with our goal to help support real-time process monitoring and our customers' quest for safer, faster, and more efficient operations. So thank you. That's the end of the presentation. Here is my contact information. If we're not able to get to your question during the next few minutes or you think of one later on, feel free to contact me directly. And with that, let's open it up to any questions, so feel free to send those in. Okay, well, here's the first question that we have. Let's talk about it. It's a good one. Do your customers typically use the IMDA system to completely replace air sampling, or do they use it in addition? There's probably not one answer that can satisfy all the possible shapes that situation could take. It really depends on the application. So thinking back to that slide we had with all the applications listed and those on the left-hand side being the more formal applications that require regulatory submission. In many cases there where you're monitoring controlled spaces, manufacturing clean rooms, etc., our customers are initially looking at using the IMDA as well as their traditional method in parallel. And they have the goal of eventually, after implementation and validation, that they could reduce or eliminate their dependence upon those growth-based techniques. They see a big cost savings there. There's a good return on investment. But both customers and regulatory agencies are not necessarily interested in trying to complete that transition immediately. They want to build up some historical data and build confidence that the data they're collecting with the new method is really giving them assurance that they're continuing to maintain a quality environment. That being said, customers are also finding that with the IMDA system, they're able to generate a whole bunch more data than they've ever been able to generate before, regardless of whether or not that's replacing the traditional method. A traditional method, maybe during an 8-hour manufacturing shift, they might collect 3 air samples, each of a cubic meter. So you have one at the beginning, the middle, and the end, and that's supposed to be used to demonstrate control during an entire 8 hours of manufacturing. Whether that can be adequately demonstrated is not my place to say, but what customers are finding is that with the IMDA system, 8 hours of continuous data can be used to support whether or not they maintain control in their manufacturing. So it's not necessarily in those cases an issue of replacing, but more supplanting the data from their traditional air samplers. And some of the non-traditional applications, those that were on the right-hand side of the graphic we were looking at, the investigations, gowning training, etc., traditional growth-based methods may or may not even be part of those applications. So it wouldn't necessarily be an issue of replacement there. It would be an issue of generating new value by using a new technique in those different applications. So it depends, but the possibility is there if the user is willing and able to take the time to generate the data to support reducing or replacing the traditional technique. Okay. So let's go to another question. How is microbial identification performed? That's a common question that we get. So microbial identification with this rapid method is not necessarily performed. It's not a genus or species identification because, as I mentioned earlier, what we're detecting is the presence of universal fluorophores, including NADH, riboflavin, and dipicolinic acid. Because those fluorophores are universal, they're nonspecific. So you're not going to be able to get a genus or a species identification. Simply the software and the instrument is going to tell you whether or not the particle is biologic or not. So if you want to do an identification, you would need to use either a traditional technique, or there are some rapid micromethods out there now that are commonly used for identification. And actually that's a good point to bring up because it does illustrate the value that traditional methods can still play in demonstrating environmental control. There is value in collecting a sample on an agar plate, and if you're able to grow it, you're able to subculture and identify it. And so that's an important piece of information when trying to diagnose the root cause of any sort of excursion you may have in your environment. But the IMDA system itself is not able to identify. So another question that we have here, are there any disposables associated with this system? Well, because the IMDA system is not using any reagents, it's not using any agar plates, there's no real disposables associated with the system. Like any piece of precision monitoring equipment, it is going to require calibration and servicing on some sort of periodic basis. But in terms of operating the system directly, there are no consumables. You can operate it continuously. You can operate it for extended periods of time. And really all you're relying on is the laser that's inside that's operating continuously to provide you those results. So it does not have any consumables. Oh, that's a good segue actually to another question that just came in. Does the device need to be calibrated? If so, how and how often? So typically the device does need to be calibrated. That is very important to ensure that you're maintaining the accuracy and sensitivity that we have validated, the ability to detect fluorescence from a single microbe, and the ability to enumerate and size particles correctly. That performance does need to be periodically checked and recalibrated. We recommend a yearly calibration cycle, and most of our customers typically adhere to that. There might be certain applications or certain environments where some customers want a more frequent confirmation that the system is operating nominally. But generally, it's a year recalibration cycle. And what is involved in that is either an on-site recalibration or shipping the instrument back to BioVigilant, where we perform a number of diagnostic tests and run a series of calibrated reference particles through the system. Not unlike the aerosol challenge that I talked about using microbes for validation, we have specific microspheres, little plastic particles that have traceable amounts of fluorescence in them. And we can run those through the system to measure and ensure the system is operating nominally. And so like I said, we recommend that that's done once per year. We have another question that just came in. How do you determine where to position the probe? That really depends on the application. It's a good question. Oftentimes, what we find is customers are going to put a probe for the IMDA system in the exact same location or adjacent to the location where they currently have a probe for either their growth-based air sampler or their particle counter. So in many environments, there are already isokinetic probes that are kind of hardwired into their critical spaces, whether that's in their aseptic isolators, the biosafety cabinets, their fill lines. Those probes are typically already positioned. And so it's simply a matter of hooking the IMDA system up with some tubing to those probes that already exist and ensuring that everything is operating nominally. If a probe does not exist, there's a little bit more consideration that needs to go into selecting the proper spot. One of the FDA inspectors that I've heard talk a lot about performing risk assessments and selecting points for monitoring in a clean room, what he always likes to say is, show me your smoke validation study. So what he always wants to look at for determining a sample location is in your critical space, what does the smoke study show? Where is the important point to monitor that would be representative of a process that helps capture any potential risk to your environment, etc. So that often means placing the probe above where your product is exposed, but not too far above, because you want to capture anything that would otherwise be deposited onto a sample. But really that's kind of almost an art that people in a manufacturing area often have specific guidelines for and already have a rough idea where they want to place it. Of course, because the IMDA system is generating data continuously and you're able to see that instantaneous feedback, one could always put a probe in an environment and then proceed to move it around over the space of minutes, hours, or days and let the data help decide where they want to place the probe. So that is a feature that is much easier to implement with something like the IMDA system as opposed to a traditional method. We have another question that just came in. Is there potential future use for this technology for assessing product sterility to replace USP71? I'm not an expert on USP71. From my understanding, there have been a few rapid methods out there that have been validated for use in sterility testing, but my understanding is that sterility testing is a requirement and there's not necessarily a way to replace it per se. That being said, I think what a lot of our customers are interested in is the parametric release that we talked about. And so in parametric release, I believe a sterility test would probably still be part of that process, but the disposition of your batch wouldn't necessarily be on hold while you're waiting for the result of the sterility test, especially if it was a traditional sterility test that required, something like 2 weeks. With parametric release, even though you're still taking a sterility test, if all of your other data, which could include data from the IMDA system, says that your environment was under control, that would give you the confidence to release, the confidence and the support, the justification to release the batch with the knowledge of extremely high probability that your sterility test is going to come back clear. But implementing parametric release, of course, is kind of a broad endeavor. The IMDA system alone couldn't help you achieve parametric release. It's a whole suite of technologies and methods that once implemented give you that holistic picture of what your process is doing and ensure that your process is under control. So I think that is all the time that we have. We've hit the 1 hour mark. So thank you very much for joining and for your questions.
Thanks, Scott, for that great presentation. On behalf of Azbil, BioVigilant, and today's speaker, thank you again, everyone, for joining, and have a great rest of your day.